Okay, hello, dear bioinformatics and genetics student. I'm going to explain the vesicular transport. I really hope that I improved my iPad capacity because I was not really good at it. At least I'm trying. So, what I'm going to try to explain here is the movement of protein within the endomembrane system. Remember, endomembrane system consists of ER, Golgi, all these vesicles, and the plasma membrane. And remember, it evolved with the evolution of the eukaryotes, because the eukaryotes are big. So, there are two, well, let's first start with the ER. Please always remember that ER is the organelle of production. So many things, including the proteins and the lipids, are produced in the ER. Of course, the proteins are produced in the rough ER, and the lipids, the phospholipids, are produced in the smooth ER, but altogether they are produced here. And from here, they are distributed to different portions of the cell. So you will know that for the cell division, the plasma membrane has to grow, but that growing cannot be only with the proteins. Of course, a good amount of plasma membrane is required, and the origin of that plasma membrane, origin of that phospholipids, is the ER. That's the first thing, and many membrane proteins are required. They are also produced in the ER, rough ER. Of course, they are not directly transported. It's transported through several pathway, which starts from the ER and goes to here. Okay, on the way, of course, it is not only transported, but it's also checked and it's modified. So from here, ER, to here, Golgi, it is checked several times. It is checked for several things. First for conformation, and then many modifications are done, and then it is brought to the membrane. Okay, the reverse is also possible. It is not always that things are produced here in the Golgi and then transported to the membrane, but sometimes things are taken from the outside environment for different purposes and taken inside and usually digested. So let's see one by one how it's happening. First, we should see how the vesicles form, because all in all, we are talking about a plasma membrane. I mean, a piece of membrane. This piece of membrane, you know that it's liquid and movable. So you can curve it a little bit, but it's not going to stay there forever because it is as liquid as olive oil. So if you want to push the membrane, you have to push it with the help of proteins. So what kind of proteins? They are mostly clathrin, COP1 and COP2. So you can imagine that with the help of the proteins, you are making a little bit of curving and more curving and more curving, and then finally it's going to be a vesicle. And we know that vesicles are now no different from the organelles. They have the same phospholipid composition, they have the same protein composition, plus they are also carrying cargo. So this vesicle is going to take up specific cargo, let's say the pink protein. The pink protein is also within this organelle. And how the vesicle is going to select them? There is no way. Because around the, which color is it? Yeah. Around the organelle, the curvature is done with some curving proteins, or let's say some coat proteins. This coat proteins has no selection mechanism. The selection mechanism is done by the receptor proteins. So there are some receptor proteins which are selecting for the targets. They are selecting for the pink proteins. And these are the receptors. the receptors and the coat proteins, again, they are not directly interacting. There are adapters between them. Adaptin, let's say. Okay. So all these cargo molecules and receptors, adaptins, and the coat proteins, they all together form the vesicle. And finally, sorry, this is what you're going to see. From the parent organelle, a vesicle forms. Like a few minutes ago, this vesicle was here in the parent organelle, but now it's here apart. Let's assume this picture, for example, it was here previously, it was part of the ER, and then it started to emerge, and then it became a free organelle. So now it's going to go to Golgi. So how is this way all goes from ER to Golgi? Let's see once again. So, of course, it is carrying proteins, and carrying these proteins is not random. It has some receptor proteins, and these receptor proteins, they are selecting for the cargo, because when you're transporting things from ER to Golgi, you don't randomly transport everything. Otherwise, ER and Golgi will be the same thing, but we know that they're not. Some specific proteins, they will be transported from ER to Golgi. And of course, there should be a selection mechanism, and this selection mechanism should work good. So, and also you might notice that we are missing two things here. The clathrin proteins are gone. Or clathrin is just one of them. It's the coat protein is gone, because the purpose of the coat protein is to curve the vesicle. And now it's anyway highly curved. And the adaptins, they're also gone. We don't need them anymore for this vesicle. Well, for another vesicle, they are going to accumulate somewhere else and make another vesicle next to it. Of course, this vesicle formation never stops. But for this specific vesicle, you don't need them anymore, so they are shed off. So this vesicle is going to go to the next organelle this way. So one of the, well, we have a nice picture here. Here you can see the coat proteins and the adaptins. And here you can see the cargo proteins. I think it was this color. So it is curving more, and more. And here you can see it from the top. And of course, you can tell the difference between these two pictures. This is TEM, transmission electron microscopy, and this is SEM, scanning electron microscopy. So this is a cross section, this one is a cross section, and this one is a visual from above. Okay, everything is nice, everything is going, but there is a little problem here. When the vesicle pinches off here, it's not that easy, because this membrane is hydrophobic and this membrane is hydrophobic. And you're trying to separate two hydrophobic things from each other, which is not easy. So you have to apply force for that. And what kind of force can you apply? Well, dynamine. So dynamine is going to move around it as if you're drowning somebody from the neck. Thank you for my wonderful drawing. So pulled from both sides. So this membrane is separated from this membrane. So it becomes a free vesicle. This is a free vesicle. So you can still see the coat proteins around it, but soon it's going to shed it off and stay with the cargo inside. Okay. Now the next thing is, oh yeah, I have a nice picture here too. The next thing is, how is this vesicle is going to know where it's going, because they are moving on the tracks called microtubules, We know that the microtubules are all around in the cell. They are spanning from the center of the cell, which is called the centrosome, to the membrane, and all the movements are done on this microtubules with the help of kinesin proteins, kinesin motor proteins. So the kinesin motor proteins are going to take this vesicle and move it on the way. But how it's going to know that if it is still close to ER, if it came to Golgi, if the membrane it encountered is lysosome or another vesicle, because of course it's going to come across to a membrane, but it doesn't know which membrane it is, so there must be a some marker. The nice thing is that when these vesicles emerges, it is not only clathrin, adaptin, and the receptor proteins, but they also carry, let's make it this colorful thing, a protein called RAB. RAB is a monomeric GTPase, which is very useful for the target recognition. And the second thing is it contains snare proteins. So anyway, when these membranes emerge, they emerge with the RAB and the snare proteins. So, like in the previous case, there are microtubules all around, and these vesicles are moving. They are coming from ER, they are moving to the Golgi, they are on the way, but of course, these vesicles, they don't have eyes, they don't have ears. So how are they going to know if it comes to Golgi or not? So it's a cooperation between both the vesicle and the Golgi. First of all, the Golgi has a tethering protein. So this protein is moving like a fishing line. It's going all around, going this way, going this way. You know, like the fishing line, it changes shape a lot because it is really looking for vesicles coming. When it comes across to a vesicle which is destined to Golgi, it's going to recognize it from the RAB protein. So RAB GTPase is kind of stamp showing the target of the vesicle, showing where it is going to go. So this tethering protein is going to take it close to the membrane, take it even closer, but again, it cannot fuse this membrane and this membrane because these two are hydrophobic, and in the middle, there are water molecules. And we know that water is hydrophilic. So these two membranes, this and this, they have to push away this water and they have to fuse. Once they push away the water, fusing is not that difficult. So what they used, the trick they used is the snare protein. So we have a snare protein already here on the vesicle. It is called V-snare because it's a vesicle snare. And here on the target, which is the Golgi, there is a T-snare. So once the vesicle come close, there are water molecules here around, and these water molecules are pushed away with the help of these two proteins, V-snare and T-snare. So first they hug each other, so they love each other, and once they hug each other, they even pull each other even more, so they are the best lovers forever. And then the T-snares and V-snares, they pull apart, they pull the two membranes together, and finally the cargo molecules are released because the membrane fuses. Of course, once the two membranes fuse, the cargo molecules will be released into the Golgi. So when this is done, two things happen. First, the cargo molecules are delivered to Golgi. Of course, this was the purpose. Second, the membrane is added to Golgi. So two things. Of course, now this membrane from here to here, it's part of the Golgi now, which means ER is a little bit of smaller now and Golgi is a little bit of bigger now. But that's no problem. Remember, ER is the production center. ER produces, that's its purpose. No problem for that. And of course, ER gets bigger because it's also getting smaller here. So here, ER gets bigger and bigger here on this surface, and here on this surface, it gets smaller. This is the way that Golgi lives. As long as production continues in the ER, it's not really a problem. Okay, and this is the botulinum. This is the bacterium that produces the botulinum toxin, which is Botox. Remember, there are many people injecting Botox to themselves. What Botox does, it cleaves these snare proteins. So if there is no snare protein, there is no fusion. If there is no fusion, of course, the transfer between ER and Golgi is disrupted, but it's not only that. So, hey, come on. So, Botox also blocks neural transmission, because we know that the neurotransmitters, they're involved in the, let's say, I need a blank page. They are involved in the axon terminal. They are waiting for the action potential. Once the action potential comes, chapter 12, remember, the calcium channels open, so all the calcium here moves in, and this calcium activates the some pathways here, which I didn't explain you. So all the neurotransmitters here are released to the synapse because the vesicles now fuse with the help of snare proteins. Of course, if there is no snare protein, no transmission, the next target cell, which can be another neuron or which can be a muscle, will not work. If the muscle cannot work, it means paralysis. The muscle is paralyzed. And this is why people who injected Botox to themselves, they cannot move the muscles in their face. That's why they look that way, without any mimic or something. But of course, this is the reason that they are going to recover, because long away in the cell body or in the cell soma, the transcription and translation continues. So the snare proteins, they are going to be transcribed from the genetic information, and they're going to be translated, and they're going to be transported to the axon terminals, to the nerve terminals. But of course, in the microscopic world, this is a very long way. So all this transcription, translation, and transport of these proteins and coming until here and reaching a sufficient level to regain the movement of the muscle takes like months. This is why people recover from the Botox action within months. Okay, all for this one. I think it's a bit too long for YouTube, and then I'm going to start, what I'm going to start with is the protein modification.